Hello everyone, my name is Ife and I'm a scientist currently working at A-Star. For the next 2 weeks of the course, we'll be looking at the next topic, RNA, and its associated functions. So why do we want to study RNA? Ever since the COVID pandemic, a lot more attention has been given to RNA. On the left here, we have a cartoon of the COVID-19 virus structure. And at its core, it has RNA as its genetic material that allows it to divide in human cells. And sequencing this RNA has also helped scientists to monitor how the virus is mutating over time and how its properties are changing. Then at the bottom here, we now have the COVID vaccines, which are the first mRNA vaccines to be approved for human use. And there is a lot of excitement about the potential of RNA-based technologies. Of course, we also know about the classical functions of RNA in the central dogma, which describes how information flows in biology. One strand of DNA serves as a template for transcription to produce mRNA, which is then translated into a polypeptide chain that can fold into its characteristic shapes to produce diverse proteins that are important for cellular function. Here, RNA itself is also involved in mRNA processing in the form of splicing, and the ribosomal RNA and transfer RNAs are required for translation to occur. In this first lecture, we'll be looking at the core concepts for RNA in biochemistry, and especially the key focus, which is always thinking about how the structure allows a biomolecule to function. And we will look at some examples of RNA function. The material from this first lecture will be mainly what you will be tested on for your final exam. In the second lecture next week, I will describe the outlook of the RNA field and some newer technologies. And I hope to share with you the excitement in the field, and you may decide to further explore this topic in greater detail in more advanced modules. Earlier, we discussed the classical view of RNA in the central dogma, where it serves as an intermediate to convert genes into functional proteins. Next, we will discuss the primary, secondary, and tertiary structures of RNA, and we will then use tRNA as an example of the structural complexity of RNA and look at its different levels of organization. Finally, we will learn about the expanded view of RNA functions, acting as genomic material, messengers, catalysts, and also regulators. We will begin by looking at the basics, the building block for RNA. You were introduced to it by Prof. Sandip in your lectures on DNA, so here we can have a little recap. A ribonucleotide contains a ribose sugar that is connected to a nitrogenous base via a glycosidic bond, and it is also connected to a phosphate group in what we call a phosphoester bond on the 5' carbon. So we start counting from the carbon next to the oxygen atom. So this is your carbon 1, 2, 3, 4, and 5. And RNA has ribose sugar, which has an OH group at the 2' carbon that is missing in DNA. The ribose sugar is not flat, and it actually puckers a bit like an envelope folding upwards. Because of the 2' group that the ribose sugar has, RNA mostly likes to stay in this 3' carbon endo position, where the 3' carbon is bent upwards in the same direction as the nitrogenous base. In this table, we have the structures of the 4 main bases found in RNA. You have adenine and guanine, which are purines with the double ring structure. So maybe a helpful memory tip I always remember purine has 2 syllables, so it has 2 of the main bases and the double ring structure. And here on the other side, you have the pyrimidine bases with the single ring structure, where cytosine and uracil are commonly found in RNA. These conventional bases can be modified, and here are some of the ribonucleosides that we will see in the later part of our lecture. So here you have inosine, which is modified from adenosine, and the 7-methylguanosine, like its name, it has this extra methyl group here. And this is what is used for the 5' cap of mRNA, which we will look at later. In some cases, thymidine can also be found in RNA, and compared to uracil, it has an extra methyl group here. Uracil, or we call it uridine when it's connected to the ribose group, can be modified in a few ways. So here at the bottom, we have pseudouridine, where there is rotation of uracil, the base here, of 180 degrees around this axis. So now it's connected to the ribose sugar via a carbon-to-carbon bond rather than a carbon-to-nitrogen bond. And finally, we have dihydrouridine, which is where the original carbon-to-carbon double bond is saturated now. So it's a single bond. So this is now not going to be flat, it's not going to be planar, compared to all the conjugated ring systems. And you can imagine this is going to disturb the stacking interactions between your conjugated rings. So the key take-home message from this slide is that conventional bases can be modified to add diversity and function. And for my part of the course, I don't expect you to be able to memorize and draw the structures of all the RNA bases, but you do need to recognize general properties and functional groups, like whether a nucleotide is a purine or pyrimidine, and the location of phosphate groups, whether something is a nucleoside or a nucleotide, so whether it has a phosphate group, and you should also know about their ability to hydrogen bond. With that, let's move into the first level of structural organization, the primary structure, which refers to the sequence of RNA bases on a strand of RNA. An RNA strand is formed by the sequential addition of ribonucleotides through a condensation reaction, where this is the 5' to 3' direction, and you have the 3' OH group attacking the phosphate group of the incoming nucleotide to form a phosphodiester bond. The presence of the 2' OH group on RNA has a profound effect on its stability. RNA can spontaneously hydrolyze because its 2' OH group can act as a nucleophile, especially if it gets deprotonated to become negatively charged, to attack the 3' phosphodiester link. And in the second part of the reaction, you have water regenerating the 2' OH group. The rate of spontaneous hydrolysis, or cleavage of the phosphodiester backbone, is faster for RNA by many million times compared to DNA, so it is chemically less stable. However, because of the same 2' OH group on the ribose sugar, in the double helix form, the RNA double helix is thermodynamically or structurally more stable than the DNA double helix. And we will revisit this when we talk about secondary structure later. There are some features that help to stabilize RNA. First, degradation of mRNA by 5' exonucleases is prevented by having a protective 5' cap. The cap structure is found in almost all eukaryotic mRNAs and is formed by adding the modified nucleotide we saw earlier, the 7-methylguanosine, to the 5' end of the mRNA molecule through a 5' to 5' triphosphate linkage. So it has 1, 2, 3 phosphate groups connecting the 5' OH of the capping nucleotide and the 5' OH of the first nucleotide. So this helps to prevent the 5' exonuclease from recognizing and binding to the mRNA. And it also has other regulatory roles. In addition, the 2' OH groups of the first and second transcribed nucleotides are sometimes methylated, and this has effects on the processing, translation, and stability of the mRNA. Another feature is the poly-A tail that is added to the 3' end of mRNA, and this occurs in a multi-step process. The transcript is first cleaved at the poly-A addition site, followed by addition of a series of A's. In eukaryotes, the poly-A tails can be 150 nucleotides long, and this tail is recognized by the cellular machinery to help in mRNA transport and also help to delay the degradation of the mRNA. So we've looked at the primary structure of RNA, which is the sequence of nucleotides on a single strand of RNA. And now we will first look at hydrogen bonding between the bases or base pairing, which helps to allow RNA to form energetically favorable secondary structures. On the left, we have conventional Watson-Crick base pairing, which is similar to the case in DNA. You have guanine and cytosine being able to form 3 hydrogen bonds in this orientation, whereas adenine and uracil can form 2. And like DNA, because the location of the ribose sugar is here, the distance on this surface is going to be larger, so this forms the major groove in a helix structure, and round this way is the minor groove. There are also non-canonical base pairing between nucleotides, including the modified ones that we saw earlier. So this is inosine, and this adds to the variety of interactions that RNA can form. For example, guanine can not only base pair with cytosine, but also uracil, and inosine can base pair with cytosine, uracil, and adenine. This allows for wobble base pairing in codon and anticodon binding, which we will go through later when we talk about tRNAs. So the extended variety of modified bases and base pairing interactions can allow RNA to form more shapes and structures and have different functionalities. Now we will look at some of the secondary structural elements that can form in RNA. When 2 regions on a single RNA strand, or it can also be 2 different RNA strands, when they can form complementary base pairs, a double helix will be formed in that region. An RNA double helix can only take on the A form because of the steric effect from the 2' OH group on the ribose ring. So like we saw earlier, this means that the ribose ring prefers to be in the 3' endo form. So this means that the 3' carbon is bent towards the nitrogen base. And this is also energetically favorable for RNA because it provides the right orientation for the 2' OH group to form hydrogen bonds. So it can coordinate with a water molecule, there's a water molecule here, to hydrogen bond with the oxygen atom in the next ribose ring here, as well as the 3' phosphate group, the oxygen on this phosphate group. Earlier, we also talked about how the RNA double helix is more thermodynamically stable And this is because it's not just the hydrogen bonding between complementary strands that helps to stabilize the double helix. The van der Waals interactions and base stacking between neighboring bases are also important. Because RNA takes on the A-form helix structure, it has a wider helix radius than the B-form DNA, and it also has a shorter pitch of the helix, so a shorter distance to complete one turn around the helix. And this means that neighboring bases are stacked closer together with stronger stabilizing interactions. RNA can tolerate the presence of unpaired nucleotides within a helical region. When the unpaired nucleotides all lie on one strand of the helix, this gives rise to what we term a bulge. Here, there is one unpaired nucleotide on this strand of the helix, so we call it a single nucleotide bulge. And notice here that the RNA backbone will end up forming a kink. Here, we have a 3-nucleotide bulge with 3 unpaired nucleotides all on the same strand. And finally, when a single RNA strand has complementary regions and can fold back on itself, but it has some unpaired nucleotides here, we call this a hairpin loop, which is also known as a stem loop structure. If there are mismatched bases within a helical region, so now there are unpaired nucleotides on both strands of the helix, we call this an internal loop. Here, we have a mismatched pair with one unpaired nucleotide on each strand of the helix. The loop is termed symmetric when the same numbers of nucleotides are unpaired on both strands. So here there are 3 unpaired nucleotides each. Or we call it asymmetric when one strand has more unpaired nucleotides than the other strand. So we've looked at 3 basic secondary structural elements: the helix, the bulge, and the internal loop. Multiple of these structural elements can interact together to form more complex secondary structures. Here we have a pseudoknot structure, which is when a single RNA strand folds back to base pair with a hairpin loop. So there are now 2 connected hairpin loops. So there's one here and another one here. So here we have an example with actual bases, and notice here that there is the GU wobble base pairing in addition to Watson-Crick base pairing. Unlike for protein structure, there actually isn't a very clear definition on what is secondary structure for RNA. So some would consider this secondary structure too just more complex, but others would consider this tertiary structure. So the exact category isn't so important, but the key idea is that we are now looking at increasing levels of complexity and organization. When the loop regions on 2 hairpins are complementary and can form base pairs, they are called kissing hairpins. And this can be 2 regions on the same strand of RNA, so intramolecular, or intermolecular between 2 different RNA strands. The hairpin loop bulge contact is when there is base pairing between the originally unpaired nucleotides that are exposed in the loop region of a hairpin as well as in a bulge. And as long as there is base pairing between at least 2 nucleotides, the structure can be stabilized even when they are far apart in sequence. Earlier, we were looking at isolated regions of complementary base pairing to form different structural units. Now, we are looking at the overall shape that the entire RNA molecule or molecules take on, how they fold over together, and how the different regions interact through more H bonding and base stacking interactions. So RNAs can form stable and interesting looking macromolecules with unique shapes and functions. Let's look at the transfer RNA or tRNA, which provides a good example of how the different levels of structural organization allow it to carry out its function during translation. At the level of primary structure, we are interested to look at the sequence of nucleotides on the tRNA strand. As there are more than 20 different tRNAs, at least one for each of the 20 amino acids, they have different sequences with some variability in their length. However, across tRNA sequences, there are 15 positions that are invariant, meaning that at the equivalent position in all tRNAs, they are the same. And these conserved nucleotides are indicated here in pink. Many of the invariant nucleotides are important for hydrogen bonding to give them their higher order shape, as we will see later. tRNAs have many modified bases, with the three most common ones being pseudouridine, ribothymidine, and dihydrouridine. These unusual bases are important because they can have wobble-based pairing to hydrogen bond to different nucleotides. We know that one amino acid can have different codons, but the sequence in the last nucleotide is variable. So having these unusual bases in the tRNA allows the anticodon to bind well to the different codons. tRNAs have well-characterized secondary structure with 4 arms formed from 4 stem loop regions. Each of these arms has well-defined interactions and invariant bases, and they give rise to an overall rigid and precise structure, which is so important because each tRNA must be correctly recognized by enzymes to have the correct amino acid added to its 3' amino acid arm region here. And this must correspond to the correct anticodon. Finally, we have the overall tertiary structure of tRNA, and on the right, we have the first crystal structure of tRNA from 1974. There are extensive interactions between the D loop and the C loop that help the tRNA to fold into its compact structure. And the regions are color-coded in the same way in this diagram on the left, as well as in the crystal structure. So we can see that the acceptor arm to be linked to the amino acid is here, and the C loop in blue folds around to interact with the D loop in white. So the tertiary structure of RNA is similar to proteins, but there is a particular overall 3D shape that is quite stable. We've just examined the structure of RNA, so now let's look at some of their different functions. So earlier we looked at the tRNAs, which are important adapters, or we can also consider them as cofactors, to bring the correct amino acid to the matching codon on the mRNA, and this must be done faithfully so that proteins can be synthesized accurately according to how they are encoded. In the same process of mRNA translation, we also have a different type of RNA, the ribosomal RNA. Bacterial ribosome has 3 different ribosomal RNAs of increasing lengths, the 5S, the 16S, and the 23S RNA, and they interact with one another and other ribosomal proteins to form the ribosome. These ribosomal RNAs are also directly involved in catalyzing the peptide bond formation. There are also several other RNAs that can act as catalysts to enhance reaction rates, and we call them ribozymes. Next, we have messenger RNA or mRNA, which encodes for proteins. However, We also now know that RNA is not just a messenger, it can also be a store of genetic material. And in the past 50 years, many RNA viruses have been discovered. Many of these RNA viruses also pose serious health concerns. Other non-coding RNAs have regulatory roles. For example, the microRNAs and small interfering RNAs, and they help to control gene expression. We'll now take a closer look at these different functions in more details. Here we will take a closer look at RNA viruses, which use RNA to store their genetic material. They do so in different ways and are classified accordingly. For example, some viruses are double-stranded, have double-stranded RNA, like your rotavirus, while others are single-stranded. The single-stranded viral RNA can be positive sense, meaning that they are just like mRNA and are ready to be immediately translated into viral proteins by the host cell. So your hepatitis C virus, as well as the COVID-19 virus, they are positive sense RNA viruses. The negative sense viral RNA is complementary in sequence to the mRNA and must be converted to positive sense RNA by an RNA polymerase before translation can occur. So it uses the RNA-dependent RNA polymerase. And because the host cells do not need this, there's no need to convert your mRNA into another form of RNA. So these polymerases, they are viral encoded and are usually a good target for drug development to make antivirals, specific antivirals. So examples of your negative sense RNA are your influenza virus, your flu virus, and also the Ebola viruses. Other class of RNA virus, the reverse transcribing viruses, they go through this reverse transcription step where the enzyme reverse transcriptase is used to convert the RNA genome into DNA, which can then be stably integrated into the whole cell genome, where it gets transcribed and translated to produce virus proteins and particles. Next, we have RNA acting as a messenger, where according to the central dogma, mRNA carries the coding information from the gene and allows for the synthesis of proteins in the cytosol. Here we'll look at the steps involved and the role of other RNAs. First have ribosomes, of which the ribosomal RNAs are major components. The ribosome first binds to mRNA, and the transfer RNAs, the tRNAs, bring the correct amino acid to the ribosome by forming favorable hydrogen bonds between the anticodon and the mRNA codon. The peptidyltransferase reaction is the main reaction catalyzed by the ribosome to add the new amino acid onto the growing peptide chain via peptide bond formation. Ribosome then migrates along the mRNA to read the genetic message and translate it into a protein. RNAs can also act as catalysts, like an enzyme, and this was first demonstrated in 1981 in the lab of Thomas Cech, who went on to win the Nobel Prize for this discovery. Eukaryotic genes contain introns, which are non-coding sequences that are first transcribed and then later removed. It will seem wasteful for the cell to expend energy and resources to synthesize these regions only for them to be discarded. However, they serve an important function, which is to diversify the possible protein products that can be produced from the same gene. In multicellular organisms, Different cells need to be specialized to carry out different functions, yet all the cells belonging to the same organism share the same copy of the genome. Through the process of splicing, the same gene can be transcribed and translated differently in different cells to carry out different functions. Hence, splicing allows for an additional layer of regulation. So with this background in mind, let's take a look at the original experiment. Their team was actually intending to find out the enzyme responsible for the splicing of the 26S ribosomal RNA from tetrahymena. And to do so, they incubated purified ribosomal RNA samples with different nuclear extracts and then ran the samples through DNA gel electrophoresis to determine if splicing had occurred to release the small 400 base pair intron. Yet they unexpectedly found that protein was not needed for splicing, as in the negative control without nuclear extract, splicing could occur as long as salt and GTP was available in the buffer. Further confirmation by using protein denaturing conditions to remove any residual proteins led to the same results, showing that the 26S ribosomal RNA has self-splicing activity. Following this initial discovery, other RNA species that possess catalytic activity were also identified, and a collective term, ribozyme, was used to describe them. This borrows from the enzyme terminology used for proteins, the idea of an RNA enzyme. Some of these catalytic RNAs, the ribozymes, act on other molecules, so a separate substrate, while others have self-directed activity, so the RNA molecule acts intramolecularly to cleave itself. And we describe this as autocatalytic. Once the RNA cuts itself, it no longer has its original structure that possesses the catalytic ability, so this limits catalysis to a single cycle. Since the first observation of self-splicing of the tetrahymena ribosomal RNA by Thomas Cech's group, other ribosomal RNA genes that were self-splicing were also discovered. So these came from other eukaryotic microorganisms like algae and fungi, and they were found to share the same self-splicing intron mechanism, so they are collectively termed the group 1 introns. So there are two steps involved in this process, which are two transesterification reactions. For this, you need the free GTP, and in the first step, the 3' OH group of this guanosine nucleotide acts as a nucleophile to attack the phosphodiester bond between the last nucleotide of the 5' exon and the first nucleotide of the self-splicing intron. So this forms a new phosphodiester bond where guanosine is now at the start of the intron. In the next step, now you have a newly available 3' OH group on the last nucleotide of the 5' exon, and this then acts as a nucleophile for the second transesterification reaction. So now this nucleophile will attack the phosphodiester bond behind the 3' end of the intron to finally splice out the intron. Slide, we have a clearer diagram to illustrate the mechanism of the first step of the two transesterification reactions. So clearly showing how the 3' OH group uses its lone pair to attack the phosphodiester bond. And now you release the 5' exon, which now has a free 3' OH that can then do the second nucleophilic attack. We have been describing ribozymes that are autocatalytic, the group 1 self-splicing introns. Now, let's look at an example of a ribozyme that acts on other substrates, ribonuclease P. This was discovered by Sidney Altman around the same time as Thomas Cech, and they were jointly awarded the Nobel Prize for the discovery of RNAs with catalytic activities. Ribonuclease P is a ribonucleoprotein containing a single RNA molecule bound to a protein. It is important for tRNA processing because when the gene encoding for the tRNA is first transcribed, it produces a pre-tRNA that has a longer 5' sequence. RNase Px to cleave away this excess region. Even though RNase P is complex with protein, the RNA molecule is the one playing the major role. RNase P ribozymes use divalent metal ions to catalyze hydrolysis of the specific phosphodiester bond in the pre-tRNA to result in the formation of a mature 5' end. Hammerhead ribozymes are small sequence motifs that promote cleavage at a specific site on the RNA, marked here in yellow, and they are called hammerhead ribozymes because their secondary structure looks like the head of a hammer. And this structure can be found either within a single RNA molecule, so this is formed by different regions on the same RNA coming together, or it can also be formed between different RNA molecules. In both cases, the cleaving structure is formed when you have a substrate strand in green, here forming a specific stem loop, and you also have an assister strand in red that is complementary to form helical regions upstream and downstream of this stem loop, and then here it also has an asymmetric loop. And the hammerhead virus is very well characterized. So on the right here, you have the representation of the crystal structure of its overall 3D structure, the overall tertiary structure. So we can see how it overall folds into this shape. The hammerhead ribozyme was originally discovered in plant viruses, and in nature this is self-acting. So from a single RNA molecule that acts to cleave on itself. But now, such constructs can be engineered artificially in the lab. So this is to design ribozymes that are more enzyme-like by adding this particular sequence motif or something similar to make a hairpin close to the region on a piece of RNA that you want to cleave. And then you can design a separate assister RNA strand that can hybridize to form the base pairing here. So we can say that these engineered ribozymes are more enzyme-like because if you provide the substrate strand in excess, you have excess substrate, you can obtain multiple turnover events, and this also follows some protein enzyme kinetics like the Michaelis-Menten kinetics that you learned about in your earlier lecture. I have a diagram describing the catalytic event for the hammerhead ribozyme. And does this look a bit familiar to you? So we saw this earlier at the start of our lecture, which is the base-induced spontaneous hydrolysis, where the 2' OH group can attack the phosphate on the 3' region. So metal cations, usually the divalent ones with a +2 positive charge, they can form strong stabilizing interactions with negatively charged groups. So they help to polarize and increase the negative charge on the atoms that they are bound to. So here in the hammerhead ribozyme, it is promoting the formation of the negatively charged O- alkoxide ion at the 2' OH group of the ribose sugar. So this enhances the hydrolysis to attack the phosphorus atom in the 3' phosphoester bond. And the presence of another metal ion at this location helps to stabilize the resulting negative charges to promote cleavage of this bond here. So at the end, there will be a cyclic phosphoester connecting the 2' and the 3' OH groups. And usually in vivo, inside cells, the metal ion here will be the magnesium ion. We have a table of some different ribozymes that have been discovered in nature. And we can see that on overall they are quite diverse with different size ranges. And quite a few of them were first found in viruses. We also see the cell splicing introns that we talked about, the group 1 intron, and we focus on the mechanism. So remember that you need a free GTP to kickstart the reaction. There is also a second class called the group 2 introns with its own specific properties and catalytic mechanism. And for these, you don't need the free GTP. You don't have to know the mechanism for the group 2 intron as part of this course, but if you're interested, feel free to read up and explore. And marked by the asterisk, we have the ribonucleoproteins. So these are RNAs that are complex with proteins. And in these examples, they are involved in the expression of protein from mRNA. So first, after your mRNA is transcribed, splicing occurs to remove the introns. And this is mediated by the spliceosome. And after that, to have functional tRNAs, it requires the action of RNase P to cleave off the extra 5' sequence. And finally, the ribosome also has catalytic RNA to complete the peptidyl transfer reaction. So in these examples, they are transacting, which means that they act on others, not on itself. And this allows for multiple catalytic turnover. For this last part, I'm going to discuss how RNAs can act as regulators. Only a very small part of our genome codes for proteins, so less than 1.5% is containing exons. And ever since the sequencing of the human genome, there has been a lot of interest to understand the roles of these other non-coding regions. So about a quarter of these have been found to code for non-coding RNA, or we term it ncRNA for short, which have functional properties either in catalysis, like the transfer RNA and the ribosomal RNA that we learned about, as well as regulatory roles. So there are two important classes of small non-coding RNAs that are both around 20 nucleotides in length. And the first one we have here is the microRNA, or miRNA for short. And this is formed from a single strand of RNA. So we have a diagram here to visualize. So let's try to recognize the structural elements. So what do we have here? We have the stem loop, the complementary helical region followed by the unpaired loop. And then at this region, we also have a bulge. And this is recognized and cleaved by a protein complex called dicer. And this generates the smaller microRNA that can then bind to and recognize a specific sequence of mRNA. So you have different microRNA sequences that recognize different mRNAs. And then there is also double-stranded RNA that gets cleaved into 20 nucleotide base sequences. And like the microRNAs, they can recognize regions in mRNA. So when both the microRNA or the siRNA binds to their mRNA that they recognize, these mRNA become silenced or degraded. So they're acting as negative regulators. Both can be found naturally, but now these siRNAs are also routinely used in the lab to artificially deplete the mRNA levels of specific genes you are interested in studying. So by knowing the sequence of the mRNA, we can design complementary regions to then knock down, we can call it siRNA knockdown of the mRNA level. Different types of non-coding RNAs, and here we can see that they are broadly clustered into related groups according to their size and other features. So sequence methods have been very helpful to understand the sequence features unique to certain groups of non-coding RNAs. And earlier we talked about the microRNAs and the siRNAs that are 18 to 25 nucleotides long, and they serve as negative regulators. They can silence genes post-transcriptionally. And the key thing is they don't need to be a functional ribozyme. They don't need to have catalytic activity to be able to affect function. Just by binding alone, they can do so. They can achieve this. And from this diagram, we can also appreciate that the different non-coding RNAs are associated with very diverse regulatory functions. For example, affecting the overall structure of chromatin, so how tightly packed regions of your DNA are, and whether they can then be accessed to become transcribed. So there are very many diverse functions, and some can be at the genomic level, so like inactivation of an entire chromosome, the X chromosome. And there is still a lot more to explore in this field. Let's tie it all back together. An RNA molecule doesn't function in an isolated manner. It's continuously interacting with different macromolecules in the cell. It can interact with DNA. So when it's first being transcribed from DNA, there is complementary base pairing between the RNA and DNA strand, and they can form a helix. RNAs can also interact with other RNAs, and like in the last example we saw about the microRNA and the siRNA, they can bind to the target mRNA to be silenced. RNAs can also interact with proteins, and in the ribonucleoproteins that we talked about, the ribosome, the RNase P, the spliceosome, the RNA component is stably interacting with the protein component. So this is where the properties of RNA come in. They help to facilitate these interactions, the kind of folds and shapes that the RNA can form that are complementary to interact with these other molecules. We also revisited the central dogma and looked beyond it to understand the various roles of RNA. So in RNA viruses, for the reverse transcribing viruses like HIV, information can flow in the other direction. So you have reverse transcriptase converting the viral RNA genome into DNA. And other RNA viruses can use RNA to make more RNA to synthesize viral proteins. So the RNA can form more RNA before it gets translated to form protein. So again, always think about how structure enables function. All these diverse roles of RNA depend on their unique primary sequences, their ability to base pair, to hydrogen bond, to then fold back on itself to form biologically functional structures that can interact, and altogether this affects the cellular regulatory state. This is all that I want to cover for this first lecture, and if you have any questions, feel free to email me at this email address here. And we can also go over other questions in the Coffee Online session on Monday. So see you!